So, as we learned in our first presentation on cellular respiration, the first stage of this chemical reaction is called glycolysis. In glycolysis, you take one molecule of glucose and you break it down into two smaller molecules of pyruvate. Cellular respiration is a metabolic process, kind of like burning fuel. It releases a lot of the energy in food to make ATP. This ATP provides cells with the energies they need to carry out the activities of life. And the reaction is C6H12O6 plus O2 yields CO2 plus H2O plus ATP. Now we move on to stage 2, or the actual production of ATP. So remember, we had glucose, it turned into pyruvate. We talked a little bit about what happens when there's no oxygen present, where pyruvate would go into anaerobic respiration or fermentation. And now we're going to focus on aerobic respiration, where there is oxygen present and the pyruvate goes into the mitochondrion in order to produce ATP. We have two further steps in this part of the production of ATP. The first step is called the Krebs Cycle. This is a series of reactions that produced energy-storing molecules during aerobic respiration. They produce a little bit of ATP. And then you have the electron transport chain, where during aerobic respiration, lots of ATP are made. A little history about the Krebs cycle. It was discovered by Hans Krebs in 1937, so in the history of science, that's not that long ago. He received the Nobel Prize in Physiology or Medicine in 1953 for his discovery, but he was forced to leave Germany prior to World War II because he was Jewish. So the Krebs cycle is also known as the citric acid cycle. In this cycle, you complete the breakdown of glucose. So you started out breaking down glucose into pyruvate. That pyruvate gets completely broken down now. Takes those 3 carbon pyruvates, breaking them down, and the carbon and oxygen atoms from the glucose end up in carbon dioxide and water. The hydrogens and the electrons are stripped off of the various parts of glucose and then loaded onto two things called coenzymes, which are NAD plus and FAD, to produce NADH, here's a hydrogen, and FADH2, another hydrogen. The Krebs cycle produces only 2 more ATP, but it loads up the coenzymes, which we just talked about, with hydrogen and electrons, which then move to the 3rd stage where the biggest amount of ATP can be produced. All of that occurs in the matrix inside the mitochondrion. So the first step occurs inside the cell. The second part occurs inside the matrix. The matrix is the liquidy part of the mitochondrion. So here we have it in picture form. Here's glycolysis, taking glucose, producing pyruvate. You get a little bit of ATP, just 2 molecules of it. That pyruvate enters the mitochondrion. You have the citric acid cycle, or the Krebs cycle, which produces a little more ATP, but it sends those electrons over to the next step. Before breaking down what happens during the electron transport chain, I want you to watch these clips of the process.
The electron transport chain is a series of protein complexes embedded in the mitochondrial membrane. Electrons captured from donor molecules are transferred through these complexes. Coupled with this transfer is the pumping of hydrogen ions. This pumping generates the gradient used by the ATP synthase complex to synthesize ATP. The following complexes are found in the electron transport chain: NADH dehydrogenase, cytochrome BC1, cytochrome oxidase, and the complex that makes transfers are also involved: ubiquinone and cytochrome C. Other key components in this process are NADH and the electrons from it.
Just so you're aware, you do not need to know the names of all these tiny little parts and pieces. Just get the overall impression of what the electron transport chain does.
Hydrogen ions, molecular oxygen, water, and ADP and PI, which combine to form ATP. At the start of the electron transport chain, 2 electrons are passed from NADH into the NADH dehydrogenase complex. Coupled with this transfer is the pumping of 1 hydrogen ion for each electron. Next, the 2 electrons are transferred to ubiquinone. Ubiquinone is called a mobile transfer molecule because it moves the electrons to the cytochrome BC1 complex. Each electron is then passed from the cytochrome BC1 complex to cytochrome C. Cytochrome C accepts each electron one at a time. 1 hydrogen ion is pumped through the complex as each electron is transferred. The next major step occurs in the cytochrome oxidase complex. This step requires 4 electrons. These 4 electrons interact with a molecular oxygen molecule and 8 hydrogen ions. The 4 electrons, 4 of the hydrogen ions, and the molecular oxygen are used to form 2 water molecules. The other 4 hydrogen ions are pumped across the membrane. This series of hydrogen pumping steps creates a gradient. The potential energy in this gradient is used by ATP synthase to make ATP from ADP and inorganic phosphate.
Remember, a gradient is when you have more substance on one side than the other, and things move, generally speaking, things move from a high amount of substance to a low amount, kind of like sliding down a slide, like we talked about before with diffusion and osmosis. So the first part of this whole electron transport chain is just creating that higher amount on one side than the other.
The ATP synthesis steps you see here are discussed in greater detail in the ATP synthase gradients animation. This animation illustrates 2 full cycles of electron donation. In biological systems, however, many electron transport cycles occur simultaneously, helping to ensure that the proton gradient is always maintained.
So the electron transport chain has electron carriers, or coenzymes. They're loaded up with electrons. Those were the bright shiny things that were in the little video. These electrons are in the form of hydrogen atoms, and protons from the Krebs cycle move to this chain-like series of steps, kind of like a staircase. You're going down the stairs. As electrons drop down the stairs, energy is released to form a total of 32 ATP molecules. Compared to the other 2 parts of cellular respiration, 32 molecules is a huge amount. Oxygen is waiting at the bottom of the staircase. It picks up the electrons, which remember are hydrogen atoms, and the protons, and in doing so, it becomes water. So here you see this in picture form again. We started back with glycolysis. Glucose turns into pyruvate, gives you a little bit of ATP, just 2 molecules. In that step, you also have some electrons carried over into the last step. The pyruvate enters the mitochondrion. The citric acid cycle occurs. You get 2 little ATP molecules formed there, and some more electrons carried by those coenzymes to the last step. The last step is called the electron transport chain, where we use these electron carriers and turn through that stair-step process that you saw in the animation, you get 32 more ATPs. So here's your energy tally for this cellular respiration process. 36 total ATP molecules for aerobic respiration compared to only 2 ATP molecules for anaerobic respiration. Which do you think is the more efficient? Doing respiration with oxygen or doing it without oxygen? Glycolysis produces 2 of them. The Krebs cycle produces 2 more. And then the electron transport chain produces 32 for a total of 36. And some say even up to 38 ATPs are made. Anaerobic organisms therefore can't be too energetic, but they are extremely important for the global recycling of carbon. We learned about carbon back in our environmental unit where we talked about the carbon cycle. So here it is all put together. Glycolysis turns glucose into pyruvate. You get 2 ATPs. You get the electrons being carried over into the last step on those coenzymes. Pyruvate goes into the mitochondrion, goes through the citric acid cycle, which produces 2 more ATP and a bunch of other electron carriers carrying those over here. You have the electron transport chain, which is like the staircase, and it takes the electrons that you carried over and turns them into between 32 and 34 more ATP. So for every 1 molecule of glucose, you get between 36 and 38 ATP molecules. That's a lot for starting out with just 1 molecule of glucose. Here's how it relates to the rest of the environment. Cellular respiration works opposite of photosynthesis. You've got the chloroplasts here, which are creating the glucose, or forming the glucose. You've got the glucose plus the oxygen going into the mitochondrion. This is where you're forming the ATP, which produces, or which powers most of the cellular work that your body needs to do. It also produces some heat, which is why you have an internal body temperature. That releases carbon dioxide and water, and that carbon dioxide and water goes back into the chloroplast along with light energy to continue making glucose. Respiration is carried out, or photosynthesis, is carried out by autotrophs. The products of respiration are used as the reactants for photosynthesis. That ends respiration and photosynthesis. This is also nearly the end of our energetics chapter, which is Unit 5. If you have any questions, please feel free to ask.